I hope you can see my.
Yeah.
Okay. I think there was an echo before. So good morning, good afternoon, good evening. Thank you for joining. I hope you can hear me
Recording in
loud and
progress.
clear. My name is Luther Gwaza, team lead for norms and standards for pharmaceuticals. And I just want to welcome you for joining us on this webinar where we are introducing the revised guideline from WHO on the biopharmaceutics classification system-based biowaivers. And I am stepping in for Steve, who for a family emergency could not join today. In case some of you are looking at the invitation that was circulated and you see that the names are different. So I'm not Steve. And my job is very simple. The first part is just to introduce the agenda and the speakers, and then we can get on to the discussion. Something happened. Again. Okay, so in just a quick introduction, I think most of you already know. Obviously, when you're joining, it means you have an interest on the topic that you are talking about. But the important thing is that generic products are essential, especially from WHO perspective, because this is one of the tools to facilitate equitable access to medicines. The other aspect that I just want to highlight why WHO is also addressing this is we have a long-standing history in terms of providing support for the development and approval of affordable, high-quality generic products. And this has been done, I think, for more than 2 decades by providing the requirements for demonstration of interchangeability of generic products with the comparator product. And we do have several guidelines, but the specific objective for this webinar is just to look at one aspect, which is relating to waiving the need for clinical BE studies. And for this, we have specific 2 topics that we are going to address. One, as I mentioned at the beginning, is to provide an update on the revised guideline on the BCS-based biowaivers, highlighting what has changed compared to the previous requirements. And this is important to facilitate implementation. So this revised guideline was just published in 2024 in the Technical Report Series 1052. And we have been doing something similar or something that is complementary to the guideline in terms of supporting manufacturers and regulators to implement these BCS-based biowaivers. And this is the Biowaiver project. So we are also using this opportunity again to provide an update on this Biowaiver project, which is done by several labs globally, essentially to carry out experiments to determine the solubility profiles for products listed on the WHO Essential Medicines list. And then also to give you an update on the list itself, so that people can utilize this list when applying BCS-based biowaivers. And we have a very good panel of speakers. Some of you are already familiar with them, and it is my pleasure to do the introduction. So first is Dr. John Gordon. John, I can see you are there online. So he is the lead bioequivalence assessor for the WHO pre-qualification team medicines for, I think, 30 years? John, no? Maybe close?
Not quite that many, but
okay.
Over 20 anyway.
Over 20 years, yeah. And he is also a senior clinical assessor for Health Canada. Again, you can see that he's extensive experience, not just with WHO, but also from Health Canada as well as ICH. So he is involved in terms of development of BE-related guidelines in ICH, but also for WHO over these more than 2 decades. And then the other speaker that we have is Professor Marival Bermejo Sanz from Spain. She is a full professor at the university in Spain with a research focusing on drug absorption, membrane transport, and predictive dissolution. She is one of the members of the steering committee on the WHO Biowaiver project, leading the work around the experiments to determine the solubility of the APIs. So thank you, Prof, for joining us as well. The other colleague that is also on the steering committee but is not able to join us today is I forgot the name just Giovanni, Professor Giovanni from the US. So he is working together with Professor Marival on this project, and he's not able to join us, but he has also contributed to the work and the material that you are presenting today. And from our side, from WHO, the focal person for the Biowaiver project is Dr. Steve. He is not available today, but he has contributed again to the materials that are going to be shared today. And if you want to reach out concerning the Biowaiver project, he is the focal person to reach out in terms of this particular stream of work. So with that, I wish to acknowledge, before we move on and hand over to John, I just want to acknowledge that this is not effort of just the presenters that you're seeing here, but this is effort of many people that have contributed to this work. So we have experts that were involved in the drafting group in the revision of this PCS-based biowaivers. We also have participants that participated in our informal consultative meetings. So some of you, I know you're already there, where they provide again feedback to this work. And then as per normal practice, we circulate our documents for public consultation. And so we appreciate also those stakeholders that had an opportunity to comment, and these comments are taken on board. And the documents were discussed at 2 previous expert committee meetings, the BCS-based Biowaiver guideline, which is 57th and the 58th expert committee on specifications for pharmaceutical preparations. And as I mentioned, the Biowaiver project is steered by a steering committee, and we do have labs across the globe that are participating in carrying out this experimental work, and we just want to acknowledge the support that you are providing to this. And then last but not least is this work could not be possible without the support from manufacturers. So essentially the data that is provided, as well as the samples for the experiments, are provided on a voluntary basis by manufacturers that are supportive to this work being carried out by WHO. So we just want to acknowledge their contribution as well. And with this, I hand over to John. But before that, we are going to take the questions at the end of the agenda. So we're going to have the 2 presentations, and then at the end of the agenda, then we open up for a question and answer. And at that point, we will, you can raise your hand. I'm sure by now everybody's familiar with Zoom. You can raise your hand and then we can give you the floor. So you can ask your question in person, but also you can utilize the question and answer chat option. And then again, our panelists will also be responding to those questions. And we can also use that platform to respond to the questions in the live discussion as well. So I encourage you to use the chat as we are going through, in case you might forget some of your questions until we get to the end. So thank you for joining, and over to you, John. I'll stop sharing.
All right. Thank you, Luther. Good morning, good afternoon, good evening, everyone. Let's see. Are we seeing my screen properly?
Yes.
Yes.
Okay, perfect. Okay, well, so to start things off today, I'm going to talk about the new WHO guideline on biopharmaceutics classification system-based biowaivers. So, first of all, I'm going to have take a couple of minutes to have a brief introduction on, and I'm sure most of you are very familiar with all of this, but on just what is the Biopharmaceutics Classification System, or the BCS, what are BCS-based biowaivers, and when are they used? The majority of my time, though, will be spent talking about the new WHO guideline and what the contents of that guideline. It talks about basically two key aspects. First of all, classification of drug substances or active pharmaceutical ingredients, APIs, within the BCS. And then it talks about the requirements for a Biowaiver based on the BCS classification of the APIs that are in a product. And as Luther mentioned, as I go through the contents of the guideline, I will make comparison the contents of this current guideline to the previous WHO guideline. And I'll also make reference perhaps to the ICH harmonized guideline on BCS-based biowaivers, which is ICH guideline M9, just because that's another important document that's out there. And so we'll take a look at how these things compare to each other. Trying to get there we go. So the BCS, it's basically a scientific framework that's used to categorize APIs based on their solubility and permeability characteristics. And I'm sure probably everyone that's in attendance here knows that. So basically within the BCS, there are 4 classes, and APIs are categorized within those classes based on their solubility characteristics and their permeability characteristics. So for example, and I'll talk about definitions of what the terms I'm going to use a little bit. And for example, an API that is a highly soluble API and is also very well absorbed, its high permeability, would be BCS class 1. So using this table, you can see how the different classes are identified. And there's some examples, of course, of APIs that fall into each of these classes on the left-hand side of the slide. So how is the BCS used? Well, it has actually many uses within drug development, right from the very beginning through to the final marketing approval of a product. The BCS classification of an API can be very important for manufacturers when they're developing their products, because the characteristics of the API will determine the formulation strategies that they need to use to deliver that API to the systemic circulation from their product. So that's the more chemistry and manufacturing side of things, but a BCS classification is also very useful in terms of determining the appropriate preclinical and clinical studies that need to be conducted to show that a API within a product is a safe and an effective product. And finally, of course, the BCS is used, and this is where the guideline focuses mainly. BCS is used to assist in regulatory applications for a drug product once it's developed, applications for marketing approval or for after marketing approval and post-marketing variations as well. So with respect to the regulatory applications, the theory is that the oral availability of an API from a finished pharmaceutical product, or FPP, can be expected to range from heavily dependent on the formulation and method of manufacture of the FPP. For example, for APIs that are class 2 or class 4 APIs, so low solubility APIs, the formulation and the method of manufacture can play a very important role in the delivery of those APIs to the systemic circulation from a product. On the other hand, highly soluble APIs are much less affected by the formulations, and they're found to be the characteristics of those products are more dependent on the permeability and solubility characteristics of the API itself. So for a highly soluble API, it can come down to the permeability properties of that API in terms of how the oral availability of the API is seen. So the knowledge of the BCS class of an API can impact the risk assessment with respect to differences in formulation or differences in manufacture between products or within a product as a product changes over its lifespan. And in fact, the BCS was originally put to use in this area in terms of exploring its uses within granting biowaivers, or waivers from the requirement to conduct bioequivalent studies for scale-up and post-approval changes or variations to products. So with respect to biowaivers then. So requirements for in vivo BE testing or bioequivalent testing can be waived perhaps under certain conditions based on a combination of things. The combination of the API characteristics, that is the BCS class of the API, but also with the considering the FPP characteristics. First of all, is it an immediate release dosage form or is it a more complicated dosage form? Whether a waiver can be granted depends on these things. And a BCS-based biowaiver requires that the dosage form under consideration be an immediate release oral dosage form. And also the FPP needs to show acceptable dissolution characteristics in order to be comfortable in waiving the requirement for a BE test on between two products or for a change within a product. So a biowaiver may be possible based on consideration of these characteristics if the risk of inappropriate BE decision is considered to be minimized by the combination of these characteristics. So when are we using BCS-based biowaivers from a regulatory perspective? We're using them in situations where you need to demonstrate BE between eligible immediate release products. So for example, and probably the most common use is when you're comparing a generic drug product to the innovator drug product, a product with clinically proven safety and efficacy. But there are other opportunities where BCS-based biowaiver can be used. For example, variations for an innovator product within its product life, or during product development prior to marketing authorization. It's possible for an innovative development program to use a BCS-based biowaiver to make a connection between an earlier version of the product and a later version of the product. And of course, later on, a BCS-based biowaiver can be used for variations of a generic product as well. But it's important to note that that's basically the scope of where BCS-based biowaivers can be used. Other pharmacokinetic types of studies, such as food effect studies, other PK-related studies, cannot be waived based on the BCS-based biowaiver. So now I'm going to turn my attention for the rest of the time, as I suggested at the beginning, to looking at the new WHO guideline of BCS-based biowaivers and look at some of the contents. Not in great depth, of course, because we don't have time for that, but we'll do an overview of the contents. And in some cases, I've got a lot of words on the slide that you can read after the fact, but we'll take a look at the contents and we'll compare, as I said, to some of the previous other guidelines that are out there. So the current 2024 WHO guideline on BCS-based biowaivers is found in the 57th report of the WHO Expert Committee on Specifications for Pharmaceutical Preparations, or the ECSPP. I am not sure if you're going to have the slide deck is going to be available to you or not, but if it is, this is an active link and you can reach it through that link, but also searching for TRS 1052 Annex 7 will take you to the guideline. It's available on the WHO website. So this new guideline supersedes previous WHO BCS-based biowaiver guidance. The previous guidance was found actually not in a standalone guideline as the new guidance is, but was built into the WHO BE guideline, the main BE guideline, which was previously found. The last version of the BE guideline that contained the BCS-based biowaiver information was in TRS 1003 Annex 6 from 2017. So from here on, we're going to take a look at the recommendations to the newly adopted guideline, and as I said, discuss the differences in that guideline compared to the previous WHO guidance, and also in brief compared to the ICH M9 guideline. Basically, a BCS-based biowaiver is a two-step process. First of all, the first step is classification of the API or APIs in a product within the BCS. So based on the aqueous solubility of the API and the permeability or absorption characteristics of the API. Once you've classified your APIs, and you then can look at the drug products themselves. So then you have to look at the FPP evaluation of the product you're interested in. First of all, and always, is it a conventional immediate release product? If yes, then it could be eligible for a BCS-based biowaiver. But other products, such as modified release products and so on, do not fall within the scope of BCS-based biowaivers. But the full FPP evaluation is basically divided into two parts. You're comparing the proposed product to the comparator product, whether that's generic compared to the innovator, or within an innovator development, for example, a variation that the proposed innovator product compared to the current currently approved innovator product. But the comparisons involve comparison of the formulations or excipient content of the two products and a comparison of the dissolution profiles for the two products. So first of all, let's look at the classification of the API, or the classification of the API within the BCS. First of all, solubility. Let's talk about solubility. High solubility. An API is classified as highly soluble if the highest single therapeutic dose is completely soluble in 250 milliliters or less of aqueous media over the pH range of 1.2 to 6.8 at 37 degrees Celsius. So, those of you familiar with the BCS will be familiar with that definition. So in order to know whether an API is highly soluble, experiments will be conducted in at least 3 pHs over the range of 1.2 to 6.8, typically in pharmacopoeial buffers, compendial buffers at pH 1.2, 4.5, and 6.8, with an N of at least 3 measurements in each of those. But also, if there's a known solubility minimum within the range of 1.2 to 6.8, then solubility studies should also, so for example, it's known in the literature that the lowest solubility point for an API is at 3.2. Then we would expect, within this context of this guideline, that the solubility study also be conducted at the pH 3.2. Once you have your solubility data over that range, the value with the lowest solubility over the range will be used to classify the API within the BCS. Basically, you can calculate what's called a dose solubility volume, or DSV. The formula for which is on the screen now. It's simply the highest single therapeutic dose, because as you'll recall, that's part of the definition for high solubility. The highest single therapeutic dose is recommended in the labeling for the product, divided by the solubility at the lowest solubility observed over the pH range will give you a DSV. So for example, if you had a highest therapeutic dose of 500 milligrams, the lowest solubility for an API, it was found to be at pH 4.5 and it was 31.2. Then you do a simple calculation to get a value of 16 milliliters. And of course, 16 milliliters is less than 250. So this API would be considered to be a highly soluble API. According to the guideline, typically equilibrium solubility experiments should be used to determine the solubility of an API over the pH range, and typically using a shake flask method, but alternative methods can be used for that. The definition of high solubility is based on a amount of drug dissolved in 250 milliliters. However, in the experimental setting, smaller volumes of media can, of course, be used to determine the solubility. And if you're conducting equilibrium solubility experiments, the experiment needs to be conducted over a suitable time frame to reach equilibrium, of course. And there's really no need for me to go into more details of that, but I should say that there's an appendix to the WHO guideline, Appendix 3, which discusses in some depth the conduct of equilibrium solubility experiments for the purpose of classifying APIs within the BCS. And I think that's a valuable tool for the guideline to provide to interested parties. The new WHO guideline, however, does provide for an alternative approach to determining high solubility, an alternative to equilibrium solubility experiments, if an equilibrium solubility experiment is just very difficult or not feasible for reasons such as, for example, the solubility may be such that very large amounts of API may be required to conduct the experiment, or there could be stability problems maintaining the pH of the buffers because of the characteristics of the API. In these cases, it is possible, as per the guideline, to demonstrate high solubility for an API by demonstrating that the highest single therapeutic dose is completely soluble in 250 milliliter volume, or a proportionally smaller amount of that dose is soluble in a proportionally smaller volume of buffer. So this is possible. This can demonstrate that an API is highly soluble across the pH range. It doesn't provide as much information, frankly, as an equilibrium solubility experiment, of course, because you don't actually have a solubility value looking at it this way. But it may be necessary for experimental reasons in some cases to assess high solubility in this way as opposed to the equilibrium experiment. The guideline does recommend and state that solubility should be established experimentally. So if you're developing a product and you want to, you have an API, you should conduct the solubility experiments so you understand that API. But literature data can be useful. to substantiate and support solubility determinations. Although you have to keep in mind that peer-reviewed articles aren't all necessarily of the same quality and may not always contain enough information to be able to determine whether the data that's presented there is suitable for making this determination for your drug substance, your API. The guideline does talk about stability too, and how stability is important in terms of assessing the solubility of an API. So adequate stability of the API and the solubility media covering the GI transit time should be demonstrated according to the guideline. So if the API is not stable, that means if there's more than 10% degradation of the API over the extent of the solubility experiment, then solubility cannot be adequately determined. In that case, you can't actually properly classify an API within the BCS. So stability of the API is an important consideration. Since we're talking about solubility determinations, I'll little foreshadowing. I'll mention, first of all, the WHO Biowaiver project, which Professor Marival is going to talk about in a few minutes. But it's basically a multi-center research project established by WHO to experimentally determine the equilibrium solubility profiles of APIs listed on the WHO model list of essential medicines. And this is a very valuable, I think, database for all interested parties, manufacturers and regulatory authorities and other interested parties. So the data collected from this project is compiled and presented in WHO reporting in the technical report series, and it's updated annually. The most recent version of the Biowaiver list is in the 2024 TRS from the expert committee, report from the expert committee. And this is the tool, as I said, that's valuable for NRAs and manufacturers and other interested stakeholders that are considering whether a product may be eligible for a BCS-based Biowaiver. But I'll let Marival talk more about that a little later. So with respect to solubility, what are the differences between this current WHO guideline and the previous WHO guideline in TRS 1003? Basically, they're very, very similar. The same criteria are used, same criterion is used to determine high solubility. So there's no change there. The previous guideline did also include an appendix that discussed equilibrium solubility experiments, so that data was available before. The alternative to using the equilibrium experiment was not discussed in the previous guideline, but it's now present in this new guideline, so there's, I guess, what would be considered an advancement. And the previous guideline didn't really discuss the importance of stability in the solubility experiments. In comparison to ICH-M9, again, the same criterion is used for determining high solubility. So there's harmonization between guidelines there. Equilibrium solubility assessment and alternatives are described. There's no appendix provided, however, with information on conducting equilibrium solubility experiments. So I think there's a strength of the WHO guideline. There is one main difference between the ICH-M9 guideline and the current WHO guideline, and is the language at the bottom of the screen here. In the M9 guideline, it says, in cases where the highest single therapeutic dose does not meet this criterion for high solubility, but the highest strength of the reference product is soluble under those conditions, the aforementioned conditions, additional data may be used to substantiate a BCS-based biowaiver, even though you're only showing high solubility at the highest strength as opposed to the highest dose. This language didn't carry forward to the WHO guideline because there was some question about the merit of it. The WHO guideline, I think, is more clear-cut on what high solubility is from that perspective. So the other important characteristic when classifying an API is permeability. There's basically three different approaches for determining the permeability of a drug product within the BCS guideline. First of all, high permeability can be concluded when the absolute bioavailability of the API is 85% or greater in an in vivo absolute bioavailability study. High permeability can also be concluded if 85% or more of an administered dose is recovered in urine as either unchanged or parent drug, or as the sum of parent drug phase 1 oxidative and phase 2 conjugative metabolites. I'll talk a little bit more about that in a second. This is data from an in vivo mass balance study. So two types of in vivo studies. But also, thirdly, permeability can also be assessed as per the guideline, but using validated and standardized in vitro methods using Keiko-2 cells. So this guideline makes a provision for using in vitro Keiko-2 systems to assess the permeability of APIs. So the test API is considered highly permeable when its permeability value is equal to or greater than that of selected internal standards in the experiment with known high permeability. So 3 different approaches to assessing the permeability of an API. Very briefly, with respect to the first of those, the in vivo absolute bioavailability study. Basically, in this type of study, you're looking at the bioavailability observed for an API following administration of an oral dose of that API compared to the bioavailability that's observed following an IV administration. So it's a simple ratio is a percentage, and if that's 85% or greater, then the API is considered to be high permeability. Now, in this perspective, this is an instance where it may be possible that literature data can be used to substantiate the permeability of an API, as opposed to having to conduct experiments yourself. So human in vivo data derived from published literature, for example, product information for the innovator product, the labeling may contain sufficient information. So this literature information may be sufficient to justify the permeability of the API for classification. Again, keeping in mind, however, that peer-reviewed articles may not necessarily contain sufficient information to properly judge the quality of the data and its applicability to making a BCS classification for the purposes of the regulatory assessment. Using the in vivo mass balance, you can again assess permeability. It's a little bit more complicated, but metabolite values can only be used in a mass balance calculation if it's known and confirmed that that metabolite was formed after absorption. So if you're going to try to figure out if 85% or more of an API is absorbed using a mass balance, then the metabolites you're including in your calculation need to have been absorbed, obviously, before they were turned into metabolites. So phase 1 oxidative metabolites and phase 2 conjugative metabolites only are formed after absorption. Those types of metabolism do not occur in GI fluids. So when you're collecting urine and feces and measuring phase 1 oxidative and phase 2 conjugative metabolites, then you know they can be included because they were formed post-absorption. Further to that, though, like for example, unchanged drug that's in feces can't be counted towards the extent of absorption generally because you don't know if that was just never absorbed and passed through the GI tract, or if in fact it had been absorbed and then excreted back into feces. So unless that can be demonstrated, the non-chain drug can't be used as part of your mass balance calculation. And any other metabolites that may be formed through pathways that could occur pre-absorption can't again be used in your mass balance calculation unless you're able to show that those metabolites are being formed post-absorption. So the mass balance study is complicated, but it is one way of assessing the permeability of a drug substance. Now the final in vitro method is in vitro methods using Keiko-2 cell systems. And this I think is a valuable tool that's easier for a lot of industry to make use of it compared to conducting the in vivo studies. The guideline provides a fair bit of information on Keiko-2 cell permeability studies, including information on method validation, assay considerations, and the list of model drugs that can be used in the in vitro studies. All of this information is contained in an annex to the guideline. But basically, evaluation of API permeability is based on the comparison of its permeability to internal standards of known high permeability. And again, if the API is showing that it's got permeability the same as or greater than those internal standards, then it's considered to be highly permeable. Stability again comes up when it comes to permeability assessment because you need to know additional data is needed to document the API stability in the GI tract over the course of a mass balance study to demonstrate that if a mass balance study is being used to demonstrate high permeability. And so the experiments that are required are described in the guideline. You need to show that there's stability of the API under the conditions that are needed. Looking at the differences between guidelines with respect to permeability, looking back at the old WHO guideline, basically the same approaches for in vivo approaches are described in the old guideline. The old guideline does make also reference to in vivo intestinal perfusion in humans as an acceptable approach to assessing permeability. That's not discussed in the new guideline. However, the in vitro method using Keiko2 cell systems is in the old guide only referred to as only supportive data. So based on the old guideline, you would not have been able to confirm a BCS classification based on Keiko2 cell data. Within the new guideline, it can be used as for classification purposes. And the old guideline, again, doesn't really discuss stability to the degree that it's discussed in the new guideline. In comparison to ICH-M9, again, the same methods and criteria are described in the WHO guideline and M9 guideline, and the M9 guideline also provides some detailed information on Keiko2 cell permeability studies in an annex, just as the WHO guideline does. So basically, there we are. So let's look at the eligibility of an FPP for a BCS-based Biowaiver. Now that we've been talking about classification of the APIs within the BCS, let's look at the BCS-based Biowaiver. So for an FPP, or a finished pharmaceutical product, to be eligible for BCS-based Biowaiver, the following criteria should be satisfied, according to the guideline. First of all, it has to be an IR dosage form, an immediate release dosage form with systemic action. That's the starting point. It has to be, if you're looking at a BCS-based Biowaiver, the two products being compared have to be the same dosage form and have the same strength. And finally, an FPP for consideration for a BCS-based Biowaiver can only contain BCS Class 1 and Class 3 APIs, or a combination of those. If an FPP is found to be eligible for a Biowaiver because it meets those criteria, then of course the FPP itself needs to be evaluated to determine whether that Biowaiver is possible. A couple of extra notes from the guideline. FPPs that are not eligible for a BCS-based Biowaiver include products excuse me, with buccal or sublingual absorption, and also products that are labeled that they can be administered without water. So for example, oral dispersible tablets can be administered with or without water and not eligible for a BCS-based Biowaiver. Because again, one of the underpinning principles for the Biowaiver is the solubility of the API in that volume of water. So if water is not involved in the product, then the principles of BCS aren't applicable to the product being considered. So at this point, let's take a quick look at the guideline and what it has to say about step 2 of the Biowaiver process, that is the evaluation of the FPP itself. So with respect to FPP evaluation, the selection of the appropriate comparator product is exactly the same principles are used as would be used for the selection of a comparator product for an in vivo BE study. Exactly the same principles are used. And of course, the bio batch, the batch of the proposed product that's going to be used for the purpose of the FPP evaluation within the Biowaiver should be reflective of the proposed commercial batch, just like the bio batch that would be used in a BE study needs to be reflective of the proposed commercial product. So those principles are the same, whether you're looking at a BE study or you're looking at a BCS-based Biowaiver. Now, the two key elements to the evaluation of an FPP are the comparison of the formulations or the excipients between the proposed product and the comparator product, and the comparison of the dissolution characteristics of the proposed product and the comparator product. So first of all, let's consider the evaluation of the excipient content of the products. Ideally, the composition of the test product should mimic that of the comparator. If the proposed product is using the same excipients in more or less the same amounts, then of course the risk that it's going to behave differently in vivo is minimized. So for that reason, that's an ideal situation. A product being developed matches the existing comparator product as much as possible. However, when excipient differences exist, these need to be assessed for their potential to affect in vivo absorption. So the characteristics of the excipients need to be considered. Do they have potential impact on bioavailability of drugs, of APIs? Is that known? And if so, then you have to pay particular attention to those. So the excipients that are known to have an impact on bioavailability of drug substances are referred to as problematic or critical excipients. These excipients have to be included in the same amounts between the proposed product and the comparator product. And by same amount, we'll talk about it more in a minute, basically we're talking about the amount of the critical excipient in your proposed product should be within 10 plus or minus 10% of the amount of that critical excipient that's present in the comparator. And some critical excipients are, for example, the sweetening agents like sorbitol, mannitol, sugar alcohols. Surfactants are critical excipients, and there's some other excipients that are known to affect the bioavailability of APIs, and therefore they're considered critical, and you have to be very careful with those when you're considering a biowaiver. So with respect to products that contain class 1 APIs, so highly soluble, highly permeable APIs, the guideline says that qualitative and quantitative differences in excipients are permitted between the proposed product and the comparator product, except for those excipients that may affect absorption, the critical excipients, which should be qualitatively the same and quantitatively similar, that is, within 10% of the amount of the excipient in the comparator product. Also, of course, if you happen to have more than one critical excipient, the cumulative difference for excipients in those products that may affect absorption should be within 10%. The guideline does conclude a little flow scheme that you can use to help determine whether the importance of the excipients. So for BCS class 1 containing active pharmaceutical ingredients, basically are the excipients in the formulation with known or suspected effects on drug absorption? If no, then a biowaiver is no problem. If yes, then you need to consider the relative amounts of that excipient or excipients in the proposed product relative to the comparator product. For products containing class 3 APIs, it's a little bit more complicated because the risk of making an appropriate decision is considered to be higher for class 3 APIs, and these APIs are considered to be more susceptible to the effects of excipients. So for products containing class 3 APIs, the excipients should be qualitatively the same and quantitatively similar as the excipients that are in the comparator product. Again, and still, excipients that are critical, that may affect absorption, have to be present within the plus or minus 10% of the excipient amount in the comparator product, just like for class 1 containing products. For class 3 APIs, because there are restrictions on excipient differences, that they need to be quantitatively similar to those in the comparator product, much more information is needed, of course. And acceptable differences for excipients are defined or delineated in one of the tables provided in the guideline, and I'll look at that in just a second. And then the guideline also provides some examples that help you determine whether differences between the proposed product and the comparator product are acceptable, and they're shown in an appendix to the guideline. This is the table that I was making reference to. So basically, it's a table that's making reference to excipients based on their function. Are they fillers? Are they disintegrants? Are they binders? And what the allowable difference is for each of those excipient functions in terms of excipient differences on a weight per weight basis. So the amount of excipient over the total core weight of the product, how much that can deviate for each excipient function compared to the comparator product. And at the top of the table, it's making reference to, of course, the critical excipients that have to, as I said before, deviate in amount by no more than 10% than the amount that's in the comparator product. Again, there's a flow scheme that you can go through for classification of products containing class 3 APIs and the importance of the excipients and what you need to consider when deciding whether biowaiver is possible. There's one more element that needs to be considered for products containing class 3 APIs, because that the deviations in the amount of excipient is based on a percent weight relative to the total core weight. It doesn't take into account some situations where the impact of an excipient in the GI tract is based on the absolute amount of excipient that's present, and it's not based on the relative ratio of the excipient to the amount of API that's present. So sometimes excipients can have an effect on GI tract situations, or for example, effect on relevant transporters, just based on the absolute amount of that excipient that's there. So since the allowable differences for excipients in for products containing class 3 APIs defined in that table is based on this core, the percent weight calculation, It's very possible for absolute amounts of an excipient between two products to differ significantly while maintaining the proportionality limits expressed in that table. So for that reason, the WHO guideline includes a further requirement to control for differences in excipients between proposed product and comparator. And it's based on to control for possible excipient effects based on absolute amount differences between products. The total core weight of the proposed product should not deviate by more than 20% from the total core weight of the comparator product. Fixed dose combination products, just very briefly, basically the same principles apply. For FDC products containing only BCS Class 1 APIs, and they should meet, of course, the requirements for products containing Class 1 APIs. For FDC products containing both Class 3 APIs or Class 1 and Class 2 APIs should meet the excipient criteria for products containing BCS Class 3. So the more conservative criteria would apply to these types of products. Again, looking quickly at the differences between the guidelines in terms of this evaluation of excipients. The old WHO guideline stated that excipients should be qualitatively the same and quantitatively similar, just as the new guideline does. However, there were a different definition for quantitative similarity for excipients existed in the previous guideline. So the relative amount of excipients present to solid oral FPPs is considered to be qualitative similar if the differences shown in the amount fall within the limits shown in a table presented in the guideline, which we'll look at in a second. The guideline did say that excipients that may affect absorption, those critical excipients, should be qualitatively the same and quantitatively similar to those in the comparator, but that similarity was not actually defined in the guideline. And the old guideline did not present any controls over deviations of total core weight of the proposed product relative to the total core weight of the comparator product. So there's an increased criteria in the new guideline in terms of total core weight differences. Here's the table from the old WHO guideline. Now, maybe it's worth just going back. If you look at the differences in the table in the new guideline, you see, for example, deviations in filler based on percent weight are allowable up to 10%. Differences in binder up to 1%. Going back to the old WHO guideline to look at what's considered similar, you see that filler differences were limited to 5%, binder differences were limited to 0.5%. So essentially the allowable differences in the new guideline are double what was allowed in the old guideline based on percent weight per weight. But the new guideline does present that control criteria on the total core weight of the proposed product relative to the total core weight of the comparator product. With respect to ICH-M9, the same limits are employed for IM9 as the new WHO guideline with respect to qualitative and quantitative similarity for excipients. The table that's used is the same, so the percent weight differences are the same. However, the ICH-M9 guideline does not have the controls on the total core weight of the proposed product relative to the total core weight of the comparator product as the new WHO guideline does. The second method of evaluation of FPPs, of course, is looking at the comparative dissolution profiles between the proposed product and the comparator product. So again, a comparative in vitro dissolution test should be conducted using one batch of the proposed product, which is representative of the proposed commercial manufacturing process for the product, relative, and then compared it to a batch of the comparator product. There are size requirements for the batch of the proposed product that's going to be used, as listed in the guideline. And of course, there is a fair bit of discussion in the guideline about the conditions for the dissolution experiments. And very briefly, I've got the conditions listed here, but they're very familiar conditions. They're not going to be any surprise to anybody, I don't think, but we'll very quickly go through them. The guideline recommends the use of either the paddle or basket apparatus with dissolution volumes of 900 milliliters or less, temperature of 37 degrees, agitation speed for using the paddle apparatus is 50 revolutions per minute, RPM. For the basket apparatus, it's 100 RPM. So these are the agitation rates that need to be used for BCS-based biowaiver dissolution experiments. You should have at least 12 units of each product in your experiment. At least the 3 buffers, compendial buffers at pH 1.2, 4.5, and 6.8 need to be employed. You may need to use an additional pH if there's a pH minimum that's known, that's not at one of those pHs, then you may need to include that in your dissolution experiments. Because we're talking about highly soluble drug substances here for BCS-based biowaiver, organic solvents are not acceptable, surfactants are not acceptable in the dissolution media. And finally, the selection, well, not finally, there's another page. The selection of sampling times should be short for a scientifically sound comparison of the performance of the 2 products. So for example, for BCS-based biowaiver dissolution experiments, you should have sampling times at 5, 10, 15, 20, and 30 minutes. Samples should be filtered at the time of collection. So unless in situ detection is being used, samples should be filtered during the collection using either a filter on the end of the sampling probe or an inline filter. Because if you collect your samples, then you take them away and go over and filter them, even if you're filtering them fairly quickly, the dissolution is continuing in your sample until such time as the filtering occurs. So for a proper dissolution experiment for a biowaiver, the filtering should occur at the time of collection. There should be evidence that the pH is maintained in the dissolution media, and so on and so on. So all these details are presented, but they're fairly standard details for dissolution experiments. But probably the critical information, of course, then is what are the criteria for the dissolution experiments? So for products containing class 1 APIs, the product should be similarly rapidly dissolving. That is at least 85% or more dissolved within 30 minutes for both products. So rapidly dissolving is 85% in 30 minutes or more. 85% or more in 30 minutes. So for class 1 containing APIs, they need to be similarly rapidly dissolving. So 85% or more in 30 minutes, and the 2 profiles need to be similar. So that's either using an F2 profile comparison, or if both products were very rapidly dissolving, that is 85% dissolved in 15 minutes, then no further comparison is needed. They're just considered to be similar. For products containing class 3 APIs, the requirements are stricter. Both products need to be similarly very rapidly dissolving. That is, both products need to show 85% dissolution or more within 15 minutes in order to be eligible for a BCS-based biowaiver. FDCs, again, the same principles are applied as applied for monocomponent products. Very quickly looking at the differences between the old guideline and the new guideline. Basically, there aren't differences in terms of dissolution requirements. Maybe less detail is provided in the old guideline, but basically you're looking at the same criterion for similarity, the same dissolution conditions are maintained. And also for the ICHM-9, the same criteria for similarity, the same dissolution conditions. Basically, the requirements with respect to compared dissolution are the same between M9 and the new WHO guideline, and very similar to the old WHO guideline. So there we have it. The WHO new BCS-based biowaiver guideline provides quite detailed descriptions of BCS classification of APIs and the assessment of FPPs for the basis of a BCS-based biowaiver. This is one of the main advantages over the old WHO guideline, is that it provides a more in-depth discussion than the previous guideline did on all the topics. But however, the conditions and requirements are very similar between the 2 guidelines. Not that much has changed, I'm sorry, but there's more discussion in the new guideline, which I think is useful. And the new WHO guideline also has very similar conditions and requirements. They're very consistent with the ICHM-9 guideline, with only a small number of differences. So there's pretty good harmonization between international guidelines from that perspective. And that's it. Thank you very much. I'll hand it over, I guess, to Maribel. If I can stop sharing. There we go.
Thank you, John. Can you hear me?
Yes.
Okay. I had to remove my headset. So thank you for that excellent presentation, and straight also on the time. And yes, I can see that there are active questions on the chat. So maybe whilst we go to the next session, you can also look at that, and then we'll come back during the question and answer. I think some of the questions might be interesting also to, respond live. And to the audience, please continue to post your questions. For the first part, as I mentioned, we'll come back at the end to address those questions. And so for the next one, I am going to do the first intro, and then I'll hand over to Professor Marival. And then for this one, we're trying something new here. So bear with me. I hope it works. We tested it yesterday, and it should work. Let's see. Yeah, I think it looks like so. Yes, okay. So this is something that we are testing because we know that our audience also includes people who are not English speakers. So we are also testing this AI feature in Zoom and Microsoft to see if this would be useful to those who don't necessarily speak English. So this is a translation in French, and please give us your feedback at the end, whether this is accurate or if this is helpful or not. So for this second part, as we mentioned, we are going to talk about the project. I think John did a very excellent introduction already on this project. As you could see from what he was presenting, the project is essentially fitting into providing data that can be useful for BCS-based biowaivers. And so just quickly, I think we already addressed this in terms of the work of WHO on bioequivalence, or to support the development of affordable generic products. And then we have the normative standards, which John already presented on. And then we have the Biowaiver project, which is providing experimental data that feeds into the list that can be used by manufacturers. And I think John also explained this very well. And just to give a bit of background to some colleagues, I know that there are many people who might be familiar with the initial work that was published in 2006. And this list included a list of APIs in the essential medicines list and their BCS-based classification. And there's something that John mentioned during his presentation in terms of, the utility of information that you'd find in peer review papers for regulatory purposes, that in some cases, or in many instances, there's not enough information that can allow regulators to determine the quality of the data that is presented. And therefore, this is one of the issues that was picked, that the initial list was based on solubility and permeability data that was accessed from publicly available information. And then later on, through the experience from the pre-qualification, where manufacturers were actually providing the data as well, and you could see data from multiple manufacturers. I think there were significant discrepancies that were observed, and then the utility of this list was then sort of questioned. So this is what you would find in the previous publication, where you had the table with the solubility, permeability, and the BCS classification, and then also some additional comments. So just to be clear, this list was withdrawn, by WHO for the reasons that I've already mentioned, that some of the classifications were not consistent with the data that was coming to light in WHO in the pre-qualification program. So please don't refer to this list anymore. This has been overtaken by events. And then to solve the problem, because having a list was found to be useful to the manufacturers and to the regulators. So the issue was, we did not just remove the list and then forget about it, but the issue was, how do we address then the problems cited with the list? And this is where the decision was made by the expert committee to then say, we could, sort of recreate the list, but using data that is coming from experimental experiments that are done based on an established protocol that can meet regulatory criteria. So this is where this work on the Biowaiver project is coming from. It is coming to address the problems with the original list. And of course, because you are generating experimental data, this also takes time. It means the number of APIs that you're doing and that are listed is also not the same as what was done in the beginning, but the list is growing as you are moving along. And obviously from WHO, the priorities are set by what is listed in the essential medicines list. So we are looking at those APIs in the same way. And then also on the APIs that are then included in the expression of interest for pre-qualification of medicines. So the intention is to make sure that whatever we are doing is a complete package that is consistent with the support that WHO is providing within the context of access to medicines. And I've already mentioned about the collaborative nature of this work, that it includes several labs that are coming from universities, national quality control labs, WHO collaborating centers. And of course, this is done in collaboration with our PQ colleagues, for the reason that we want whatever is coming out to meet the same regulatory standards, so that this is acceptable by regulators. So this is essentially important for this particular project. So you'd see John mentioning about the protocol. So the protocol in the guideline is also what is being implemented in this project as well. And again, John mentioned in terms of how BCS-based biowaivers are applied, that there are two main things. One is the API classification that needs to be done first, and then you have now the FPP evaluation. And so the project is only looking at the solubility, which is one element. So we are not yet looking at the API permeability. So the requirements that John already mentioned in the guideline is what should be followed. And then for the solubility, for the APIs that already are included in the Biowaiver project and that are classified, then that data can be used. So what can happen then if you don't have that? It means manufacturers can still follow the approach to determine solubility in the guideline, and then provide that data to the regulators, and then the regulators can determine, whether they can accept, a Biowaiver or not. And then so the other elements remain as described in the guideline. They're not covered in this project. Just to provide an example in terms of what actually came out of the Biowaiver project, I did mention that initially we were looking at that list from 2006. And then what we are also doing is to generate this data and then look at the previous classification. So this is ethionamide, which is one example where the initial list based on literature is contrary to the data it helps to actually have concrete data that can support scientifically driven decisions on the APIs. So one classical example here, which is important to note. And with this, I think the next slide would be Professor Marival. So I hand over to you, and I will continue to move the slides on your behalf.
Yeah. Okay. Thank you very much, Luther. Well, essentially, I'm going to briefly summarize the protocol that was developed in order to provide guidance to the laboratories to design and conduct the equilibrium solubility studies in order to classify the APIs within the BCS system. So, I mean, this protocol was developed and discussed previously to provide an harmonized approach when the laboratories are performing the solubility studies. Next, Luther. So essentially, the idea is that the API sample has to be suspended in the adequate buffer that I will describe later. after and time separated by appropriate methods. And then the API concentration should be measured using a suitable analytical method. This is briefly the idea. Next, Luther. So as John has mentioned, an API is considered to be highly soluble when the highest single therapeutic dose is soluble in 250 milliliters of less of the aqueous media over the pH range of 1.2 to 6.8, meaning that the highest single therapeutic dose is the total amount of API that is administered orally at once. In general, the pH solubility profile of the API should be determined at 37 degrees in an aqueous media, and at least at these 3 pHs, as John mentioned, and at least we're using 3 replicates. Next. The shake flask method is for solubility determination is the recommended one. The idea is to use, if possible, a mechanical agitation device, and of course using the appropriately validated analytical method. The device should be capable of maintaining the temperature at 37 Celsius degree, and with, let's say, appropriate agitation speed to ensure the weighting of the particles and the particle contact with the buffer solution. In principle, we expect that with the adequate agitation, and in generally the equilibrium should be reached within 24 hours, but actually we are taking samples for a longer period of time. Next. When developing the protocol, we discussed some issues that could happen when you are performing a solubility study. For instance, that the API has poor wettability or has a tendency to float, and in that case, some solutions are allowed, like the use of glass microspheres or agitation or sonication in order to ensure that when you put in contact the API with the buffer, all the particles are wet. But the use of surfactant is not allowed, as the presence of a surfactant would bias the solubility determination. Next. Another, and next slide, Luther, please. Another potential issue is that actually, if we are working with a highly soluble substance, in order to have a true solubility, the measurement of the equilibrium concentration in the presence of an excess of solid, sometimes we will need a huge amount of solid of API in order to measure the true equilibrium solubility. I will discuss later how we address this issue. Another potential problem would be a very long equilibration time. As I mentioned, in principle, we expected 24 hours for equilibrium, but we are using longer sampling times. And the third issue is the poor API stability during the study, and we have also addressed this problem. Next. So in order to address the problem of requiring very high amounts of API in order to measure the equilibrium solubility, the alternative solution was to report what we call the lower solubility limit. I mean that the actual solubility is greater than the reported value, but this reported value is still higher than the solubility required to dissolve the highest single therapeutic dose in 250 milliliters of medium. So in this case, in the selected volume of media to perform the solubility experiments, we ensure that at least the dose, and in general a higher amount, let's say 1.5 times the dose or twice the dose, is soluble in this in the selected volume. Next. The buffers for the equilibrium solubility determination are the pharmacopoeia ones. I mean, we are using the recipes from the pharmacopoeia, and we are measuring the pH at the beginning of the experiment when we put the API in contact with the buffer, and at the end of the experiment. So we control that the pH has not changed. And if there is some change in the pH, it could be adjusted using small amounts of acids or base, depending. And if there is not possible to maintain the pH, it could be possible to use higher buffer strength in order to keep the pH control. Next. The solubility determination is made into phases, into steps. There is a preliminary assessment in order to know the time we need to reach the equilibrium. So for these preliminary experiments, ideally we use 30 to 40% excess of undissolved solid. I mean, we of course have some preliminary estimation of the solubility, some idea that you can find from databases in open sources on internet, or estimations of qualitative definition of the solubility of the substance. I mean, with this preliminary idea about the solubility, we estimate 30 to 40% excess, and we put that amount of API in 5 milliliters of buffer, for instance, at the 3 pHs. Take samples at 2, 4, 8, 24, 48, and 72 hours. And we determine the dissolved concentration in order to generate this plot of concentration versus time, in order to have an idea of when we are reaching this plateau. I mean, the two consecutive samples are not deviating significantly, and then we select the time for the next experiments, what we call the time to equilibrium. Next slide. When we're doing the experiments, filtration is the recommended media in order to separate the solid from the fluid, but centrifugation is also a valid alternative method. If you are using a filter, for example, a pore size of 0.45 microns, immediately after withdrawal the sample, or take the sample and immediately perform the centrifugation. Next. We have also in the protocol some recommendations for the analytical methods. Ideally, you should construct a calibration curve with 5 to 6 standards, and 3 additional standards in order to estimate the precision and the accuracy of the method. In general, in the run of the analytical samples, the controller intercalates with the samples. And for instance, in order to check the filter influence, the control standards could be injected without and after filtration in order to ensure that the recovery is almost complete, that the filter is not retaining the compound. And well, with this calibration curve, we determine the usual parameters to ensure the quality of the method, like linearity, accuracy, precision, etc. Next. Well, in the next phase, in the second phase, when we already know the time to equilibrium, we perform let's say, the pivotal, the definitive experiments at the 3 pHs, but we already know when took the sample. In this case, we use less excess of solid, in general 10% of excess of solid, and the procedure is exactly the same, except wait until the sampling time, keeping the samples under agitations, and then separate the solid by filtration of centrifugation, record the pH value, the initial and final pH value, and we recommend in general to dilute the sample immediately after filtering of centrifugation to avoid the precipitation and determine the concentration of the API in the sample. This is the table that is recommended in the protocol in order to present the results. As you can see here, for the preliminary solubility experiments, we have the initial pH, the final pH after correction, the amount of acid or base that you have you needed to control, to adjust the pH, which is the final API concentration, the mean of the 3 replicates, the coefficient of variation in order to ensure that the variability is acceptable, and the concentrations at the different sampling times, up to 72 hours, in order to determine the time to equilibrium. So this is the first phase of the experiments. Next. And then the final tables showing the results of the pivotal experiments. The same thing, the 3 pHs, any potential additional pH that may be required, the 3 replicates, the final and initial pH values. In that case, we also indicate how much API have we wait for the experiment and the buffer volume that we are using, and the 3 replicate results, the average, and the coefficient of variation. Next. So based on these results, we can make a representation of the pH solubility profile with the variation, with the deviation, and then we estimate, as John has mentioned, the dose solubility ratio, in order to check that this dose solubility ratio is less than 250 milliliters. It has to be less than 250 milliliters at the 3 pHs in order to consider that the API is classified as a high solubility substance. Next. I mentioned before that we also consider that there could be some stability issues that are relevant not only for the experiment, but also because the API should be stable in the gastrointestinal system. So in order to ensure that the API is stable, during the experiments, we use the same approach that has to be used in order to demonstrate that the API is stable in the gastrointestinal system. So we perform a separate set of experiments with some excess of, actually we perform them with an amount of solid that we know is soluble. I mean, we perform the stability experiments with a solution of the API, and we put this solution in contact with, let's say, acidic fluid in acidic conditions using a experimental time of 1 hour. That is more or less the average time the API is in contact with the gastric fluids. And then we measure we know the initial concentration, we measure the final concentration after 1 hour in contact with the buffer at pH 1.2. And in the same way, we put the API, the solution of the API at pH 6.8, representing the pH in the small intestine. And we incubate the samples during 6 hours, again, 37 degrees. And we know the initial concentration. We measure the final concentration to ensure that is no more than 10% of degradation on those conditions. And I think this was my final slide. Thank you, Luther.
Thank you, Prof, for the presentation. And then I quickly just conclude on this part. So as mentioned, we do have a number of labs, and then you can see where the labs are coming from, which is all over the world. And then this is what you would find in terms of the selection of what you include in each cycle when we're doing the experiments. And then we also look at the EML, but also the expression of interest for pre-qualification. So we try to balance the two as well. And then lastly, as we mentioned, you do have the support that is coming around from all these different labs, which I already acknowledged at the beginning. And then I'll just move on to the last part. So the annex or the list John alluded to, you also find it in each TRS almost annually. When we update the list, we publish the list as an annex in the new TRS. So if you want to know where to find the latest list, please go to the 2024 TRS. That's where you'd find the updated one. And then, of course, based on the work that has been done in this year, we'll update and publish another one in 2025. So on an annual basis, we are growing the list as well and then publishing it. And this is what you'd see in that list that we are publishing. This is the last slide, just highlighting in terms of numbers, what has happened since the project started. And you'd see 16 labs, 38 manufacturers that have contributed to this Biowaiver project. And then as of the current date, we have 50 APIs in the EML that have been gone through these experiments, and 300 samples received from these manufacturers. And then across all the labs, 1,500 experiments have been done. And this is over the past 6 years. So I already acknowledge the work that has been done by all these colleagues that are contributing to this important work. And I want us to move then to the Q&A. So with that, I know that John has already been looking at some of the questions. So I'll stop sharing my screen here. And then for the sake of time, John, I think you have looked at the questions, so maybe you can take them maybe one by one and already explaining, instead of me looking and then asking you. I think we've already looked at the questions, and then we can see if people want to raise their hands.
Okay. So are you seeing can you see the questions on the screen now? Am I sharing?
I can see the slides.
You can't see the Q&A.
Well, right now you are showing the slides with the BCS classification.
Okay. I thought when it showed the active screen, it would show what was ever on top of it. I don't know. Do we have a way to show the Q&A on the screen at the same time, or no? Maybe not.
Let's see. I don't think so, because it shows when I open it, it just shows on my screen.
Okay. Well, I guess the alternative is if people can pop open the Q&A. What I propose to do, because time is fairly short, we might go a couple of minutes over. I'll very quickly read the question. And most can be, I think they're worth answering live, because I think they're questions that a lot of people would have.
What are you doing?
So if people can open the Q&A and follow with me, then I'll just go through them. There are a couple that Mary Val should answer, and one or two maybe you should answer, but I'll, for the most part, just go through them, if that's okay. So the first question is asking, may I waive a BE study and claim BCS-based biowaiver if 1, my formulation is an oral solution, 2, the API is BCS class 1 or 3, 3, the percent release in all 3 dissolution media is above 85% in 15 minutes, and 4, my reference product is immediate release solid oral dosage form. Well, for BCS-based biowaiver, one of the requirements is that the dosage form being used, being compared, has to be the same. Technically, the answer to your question is no. You can't compare an oral solution to a solid oral. However, if the innovator product has an oral solution as well, you can certainly get a biowaiver based on the principles of aqueous oral solutions, because of course for an oral solution the API is already in the solution, so that's a major step. But in terms of BCS-based biowaiver, the answer is no, because an oral solution can't be compared to a solid oral. Next question, is there a scientific reason for the choice of 250 milliliters in solubility determination? Well, scientific, I don't know, but the answer is 250 milliliters is chosen because that's a typical glass of water. So, when you're taking a solid oral dosage form, you drink a glass of water, it's somewhere in the range of 250 milliliters. And that I think is the premise for the selection of 250 milliliters for the solubility experiment. So if your drug is going to completely dissolve with that glass of water, then we know it's in solution in the stomach. And I think that's the basis. Next question for you.
And maybe John, I think there's one person who has raised their hand, so maybe we can take their question.
Sure, if you can make
can you unmute them? Christina, you can go ahead and ask your question. Okay, maybe they are not unmuting. You can continue.
Okay. Well, the next question is the question of whether the record's going to be shared with participants. So I can't answer that.
Yeah. That one, yes. We'll share the record and the slides. Yes.
Please share the new guideline by email. Again, I don't know if general link can be made available or something. Is there sorry, Luther, go ahead.
I see now she has unmuted herself. So Christina, go ahead. Can you go ahead and ask your question? Well, okay, John, let's continue.
The next question is, there a specific time period over which the DSV studies are conducted? Now, perhaps this question was asked before Mary-Val spoke, so perhaps her presentation has addressed it adequately. But I don't know, Mary-Val, did you have something else you wanted to say in response to that?
I don't see the question. Could you repeat?
Oh, is there a specific time period over which the DSV studies are conducted?
Oh, yeah. I mean, I already say that in the preliminary experiments, we determine the time to equilibrium, and the pivotal are performed at that time to equilibrium. Okay. Yeah. I think it's been covered. The next question. In case the generic has different strengths as compared to the comparator, can this be accepted for biowaiver? For example, 5 milligrams generic versus 10 milligrams innovator. And the answer to that would be no. As per guideline, the same dosage form and the same strength needs to be considered for BCS-based biowaiver. So the innovator has 10, then only a 10 milligram generic could be considered for BCS-based biowaiver. Next question is asking about slides being shared, and I assume the answer to that is yes.
Yeah, that's yes.
How does the applicant quantify the excipients of the reference product? And this is a good question. What data are expected during submission to verify that the quantitative composition presented for the reference product is accurate? Well, this is always an issue. So for a product containing a BCS class 3 API, you need to be quantitatively similar to the comparator. So you need to know how much of the excipients are present. In some limited cases, quantitative amounts are available in labeling, but for most cases, only qualitative excipient content is provided. In that case, basically, the manufacturers have to reverse engineer the comparator product to develop a breakdown. You know qualitatively what's there, you need to reverse engineer to figure out quantitatively what's there. The regulatory authority, of course, will know the correct answer. So they'll tell you if you're right or not, but they won't tell you where you're wrong if you're wrong. But that's basically what has to be done.
Yeah. And then I see there's Saida. You can go ahead and ask your question.
Hi, everyone.
Hi, please go ahead.
I have quick 3 questions about the comparative dissolution profile.
Okay.
The first question, should the results of Biowaiver of comparative dissolution profile be covered by the validation for dissolution? The second question, when we use statistics, can you hear me, please?
Yes. Yeah. When we use statistics, that's the last I heard.
Or boot models for the results. The final question, if the media in the QC test is 500 or 1000 milliliter, should we use only the 900 millimeter? Thank you.
Okay, so I'm not sure I got all of the middle question, but the first question of validation. So the media that need to be used for BCS-based Biowaiver are defined in the guideline. The quality control media that's being used for your product may or may not be the same as one of those. So of course a method has to be validated. If there's media differences, then if your method of detection is UV, then you'll have to validate the method for all the media. If you're using HPLC for analysis, then perhaps the validation you've used for your QC media may be sufficient. That's a bit of a review issue. You asked something about statistics in the second question. I didn't quite hear it. But the guideline does define F2 calculations. F2 calculations should be used to compare the similarity of 2 dissolution profiles if they're not both very rapidly dissolving. So there is more information on that in the guideline that I didn't try to cover today. And I've forgotten the third question. Oh, volumes.
Yeah.
Yes. Yeah. So the guideline requires that you use 900 milliliters or less. If your QC medium uses or method uses 500 milliliters, it would be acceptable to use that in your BCS-based Biowaiver if you wish. 1,000 milliliters is about more than 900, so 1,000 milliliters should not be used for BCS-based Biowaiver experiments.
Okay.
Thank you, Doctor.
Okay.
Yeah. Because of time, John, I'm going to use my being the moderator to selectively pick questions.
Sure.
And then the other questions, all of the questions I see, there's a lot of questions. The panelists, just for the audience, the panelists will respond, both John and Marival, and questions to WHO will respond, and we will share that along with the slides. The recording will also then share the question and answer, the questions that you have provided and the responses as well, because we won't be able to cover all of them. So I'm picking one is for John. You mentioned CACO2 for in vitro permeability test. There are several commercially PMPA membranes in study. How do you think about these membranes?
So there are certainly different cell systems that can be used to determine permeability, and some of them are probably very good and perhaps even better than CACO2 experiments. However, as per the guideline, currently only CACO2 cell experiments can be used for permeability assessment for submission to regulatory authorities. And in part, it's because of the familiarity of the regulatory authorities with that system and the experience they have with that system, so they're better able to evaluate that. So what I would anticipate is, as time passes and other systems become better known and better understood by the regulatory authorities, they may then be accepted. But currently, only CACO2 systems are accepted.
Thank you, John. And then for Marival, there is a question: what is the appropriate agitation speed should we use for the solubility test?
There is not a definition in the protocol about the agitation speed. In the protocol is indicated that this should be enough to ensure the weighting of the solid and avoiding, if possible, the vortex formation in order to avoid, bubbles coming into the fluid. So let's say enough to ensure the weighting and not too much in order to avoid the vortex formation.
Mm-hmm. And then maybe another question to you as well. What information do we get from the solubility data performed at pH of lowest solubility of the API if within the physiological pH range? Any relevance to BCS classification of the API?
Sorry, I didn't catch exactly.
So I read the way it's
Yes.
expressed, but I'm going to interpret, and John, and you can help me. I'm assuming the question is, what information do we get from the solubility data performed at pH of lowest solubility of the API if maybe within the physiological pH range is different from that one? What would be the relevance for BCS classification for performing the solubility at the pH of the lowest solubility, which is outside the physiological pH range? I'm assuming this is what the person is asking.
Yeah, well, if the lowest solubility value is outside the physiological range, we don't need to know this value, because for the solubility estimation, we are using the minimum solubility value within the physiological pH range.
Mm-hmm. Okay. Let's see if we can take one more person. John, I know you were looking at the question. If there's one more interesting question, you can go ahead. I'm just looking if there's any. There are a few questions around Kako 2. I think you've already answered that.
Yeah, there are a couple I'd like to say something about very quickly.
Yeah, go ahead.
Our specialized products like solid dispersions, those that show high solubility and permeability, are they applicable for BCS-based biowaivers? The thing about a solid dispersion is it's usually only used if you've got a low solubility API, and it's not the solubility that you're seeing coming from the product. It's the solubility of the API in its own right that is important for BCS classification. So typically, if it's a solid dispersion, it's a low solubility API, and it wouldn't be applicable for a BCS-based biowaiver. So I think that's important to note. So typically, we're looking at conventional immediate release solid oral dosage forms.
Mm-hmm.
Something I thought maybe you could answer if you had just a second. There's a question, I don't know where it is now.
Mm-hmm.
Why is there a WHO guideline when ICH-M9 exists? I think that's...
I saw that one. Okay. Yeah. So that's an interesting one. Yes. So ICH, as you know, only covers specific countries that are members of ICH. So it does not cover the rest of the world. And then you have the rest of non-ICH countries that are not necessarily looking or referring to the ICH. And therefore, for that reason, WHO also looks at updating its own guideline. The second one is, for this particular topic on generic products, as you know, WHO has been publishing these guidelines and many people were referring to them. And ICH only started now to also publish harmonized text for related to generic products. And then the last comment is from WHO side, this is publishing normative standard is one of the mandate from member states. And for that process, we have to take into account the applicability of the guidelines across all WHO member states. So as you could see, the text is harmonized, as John was doing, explaining the similarities with the ICH, but also you also noted that there are some few differences. And we tend to do that to try and address the applicability of these requirements across all the WHO member states. So that's what I can say about this one. In some cases, you'd find that WHO does not go ahead and have a separate guideline. We refer to ICH, particularly for safety and efficacy data. In some cases, the expert committee and the member states would feel that it's important for some topics to have a WHO, even if an ICH guideline exists. So it's taken on a case-by-case basis. So I know we're out of time. We have exceeded by 9 minutes. And maybe to John, any last comment, thoughts from your side, and then also Professor Marival before we conclude.
No, my last thoughts would be that the questions are very good questions. I think a lot of them are answered by considering the fact that you need to have the same dosage form, the same strength, when you're looking at a BCS-based biowaiver. That addresses many things. And the dissolution conditions for BCS-based biowaiver are those defined in the guideline. And Kisu media and compendial dissolution methods don't apply.
Yeah. So essentially,
that covers a lot of questions.
Yeah. So essentially for people to follow the guideline, that the
Yes.
guideline in many cases already answers these questions. Yeah.
That's right. Yes.
Yeah. Thank you. And then Professor Marival, any last thoughts, comments from your side?
Well, just to say that in our experience, the protocol help us to have an harmonized way to determine the solubility, and even if we cannot avoid the inter-laboratory variability, what we have to ensure with the protocol are that the conclusions were always consistent across laboratories. So in general, I think this is the major outcome of the protocol, that our results has been consistent across different laboratories. I mean, the outcome, the conclusion of high or low solubility.
Yeah, and also I think what is important as I conclude is that the work of the Biowaiver project also provides feedback, because you do have this protocol also in the guideline. So I think this also helps to provide feedback so that the users, particularly for manufacturers, it's something that has been tried and tested, and we know that it works. If there are issues, those issues are also addressed. So thank you both for the excellent presentation. And again, I'll just reiterate that this is an important topic in terms of access. And also this is an important topic, I think, in terms of harmonizing. So that's why we are also taking the initiative that when ICH publish the guidelines, we also look at the WHO ones and then update them as necessary. So thank you. And then thank you everyone for the excellent comments and questions. As we have promised, the recording, we are not there yet. We have a number of other recordings from previous webinars. The intention is this should be available on the WHO YouTube channel. And then the second one is what we can circulate immediately, maybe by next week after the meeting, is we'll share with you the slides, for you to refer to them. I know people were asking about the link to the guideline. We'll also send that. And also John mentioned in his slides, this is also an active link as well, but we'll also send that along. And then the last one is we'll work with John and Professor Marival to answer all your questions and put them nicely into a document, and then we will also circulate that to you as well. So thank you for the comments that you have provided. For the sake of time, we could not go through all of them, but we'll still provide written responses. Otherwise, thank you everyone, and cheers. Bye-bye.
Bye.
Recording stopped.